Yeah, okay, thanks for this very nice introduction. And thanks to the organizers for giving me a chance to talk to you here. I'm going to say something very different from what's been said so far. One, because I'm going to present an experimental view. I'm an experimentalist, and so I'm going to be showing you a lot of videos and experiments and trying to convey to you what this active is, and it's no longer evolvable. I thought we could save that for later. I'll just tell you something about that's evolving, and also as Rajesh mentioned yesterday, GEB has always been Gödel, Escher, Bach, and so I thought I'll start with Escher, and there are games, epidemics, and a lot of behavior in this. But what I wanted to convey using this is that my interest in the kinds of things I do stems from this emergent phenomena that we see around us, and in some sense, the kinds of questions that I ask and also will tell you today about will involve stuff like how do individual constituent elements give rise to emergent properties of the system, sometimes which is quite surprising and unexpected, right? And also I want to preface this talk by saying that it's not really going to be a tutorial in the sense that I'm going to show you a continuous train of thought, but it's going to be more a smattering of things related to this aspect. In the first section, I'll try and tell you about active systems, and then in the second section, I'll tell you something about evolution. And also, mostly, I'm going to be talking about bacterial systems, yeah, let's see how it goes. So I wanted to start here because I advertise this thing as a physics perspective of biology. I got this quote from Richard Feynman in his lectures where he says, physicists always have the habit of taking the simplest example of any phenomenon and calling it physics, right? So I'm afraid some of the things that I'm going to be telling you about biology are in this spirit, in the sense that I am definitely going to be looking at simple enough things and telling you something about those phenomena, but I want to say that biology really is quite like that in many ways, right? Even though we think that there should be an elegant and a simple way for biological systems to function, it's more often not the case, and this happens to be, in many instances, because these systems evolve, and evolution somehow puts these in a sloppy domain where things just work somehow, and eventually, probably given enough time, they will reach some physical limits or optimality. For instance, we are able to detect single photons with our eyes and so on, but in most cases, whenever you look at some complicated gene network or even ecological interactions, it will quite often be something like this. And so the goal is, one, to get the simplest examples from these very complicated processes that go on, and two, we should also step away somehow from this habit and really try and understand biology for what it is, right? So these are the two main things I wanted to say. Okay, sort of a sort of a crash one-slide course on how I view biology. So there's a genotype which is carried by the DNA, and there is a phenotype, and an organism sits in the space of various traits. For example, the size of the organism, the speed at which it moves, and how it interacts, how it communicates with another individual, and so on, right? And the link between this phenotype and genotype is that there is a genotype that dictates this phenotype in some way, in combination with the environment that this organism finds itself in, and together with the environment, the phenotype feeds back onto the genotype, what I'll probably loosely refer to as evolution, right? So we are really interested in unraveling this mechanism that takes us from one to the other, and these mechanisms are of various sorts, right? And I'm going to be focusing mainly about the physical aspects of these rather than biochemical and so on. Okay. Again, there are so to underscore the point I just made, Darwin said to us, gave to us this very nice and beautiful theory of natural selection, heredity, and so on, way before genes and the DNA and so on was discovered. But all of those are just played out by the genes and the DNA, the ideas that he outlined. And all of you very well know what that entails. I'm going to be telling you something in the spirit of another famous thesis that came out around the same time. D.R.C. Wentworth Thompson wrote this remarkable book on growth and form, where he argued that mechanical forces and physical constraints play a large role in shaping organisms, how their body structures come about, for instance, or how they even interact with each other, and so on. Right? So it is in this spirit that I'm going to be telling you about active matter, because it's all about mechanical forces. Before I go further, I want to point out to you what I mean by this, or what Darcy Thompson meant, right? So this is an image of some cells in the retina of the fly eye, right? And you will immediately see that these cells are organized in a way which is very similar to what you would get if you take bubbles, soap bubbles, and put them together, right? So the pictures on here are the actual cells. These are soap bubbles which are constructed by hand on the table, right? So there's a remarkable similarity between these structures, and so the argument here, or the point here, is that one need not invoke a genotype and so on to get structures like this. There are definitely, or clearly, there's a role for things like surface tension and the forces between, or the adhesion between these two different cells, which gives rise to this shape, right? And so this is a static picture, but then all of these things come through dynamics, and this is a beautiful example of the dynamics in which both the genetic regulation and these mechanical forces play a huge role, right? So the movie that I'm going to show you now is the development of a fruit fly embryo, right? So this is the egg of the fruit fly. It's about 0.5 millimeter in size, and each of these spots that you're looking at is a fluorescently glowing marker that is in the nucleus of the cells, right? So there are many, many, many cells which sit as a sheet. So you can think of this as a balloon, right? So there's a balloon, and this balloon is comprised of each of of individual cells, right? And as soon as I play the movie, you will see that there are large-scale flows that come about, and you will also see some patterning, right? And I'll talk about all of that. I wanted to point out that to you before I played the movie. Okay, so this entire thing is happening on the time scale of hours. Okay, so what you saw, so you already saw a large flow or deformation event that happened. Now there's something else happening. There's already some segmentation that you can see over here. These stripes eventually define the spinal column or the vertebrae of the fly, right? And so all of this patterning is governed by regulation or a gene network regulation that's happening from below, where the cell is using information of the boundaries and then precisely finding the various regions along its body, and somewhere afterwards, one of these sides will become the head, one will become the tail, and so on, right? So it's not just this patterning that's coming about because of this signaling that's happening at the gene network level, but there's also a role for the flow that's going on here, right? Yeah. Exactly. That's the point I'm trying to make, that there is indeed an interplay, So while one may argue that even in the face of some stochasticity, these gene regulations are quite tightly regulated, meaning that they give rise to, a precise amount of some protein which then governs something else. But there's a large sloppiness that comes from these mechanical flows, right? So somehow both of these have to be countered, eventually giving, so every fly comes out fine with a head, with so many vertebrae, and so on, right? So there's this large role that's played by both mechanics and this gene regulation. Both of them are intertwined, clearly, right? And the goal somehow of this program is, of this large research program, is somehow to figure out what that link is, right? In some ways, okay? So these, yeah. Exactly. So the morphogen gradients are what creates those patterns, right? So, and exactly. Exactly. Right, exactly. So there is some other role for the flow here, and I won't go into the details of this itself. I just want to point out to you that, there's both mechanics as well as the kind of processes that you're talking about, right? So for instance, there's some, yeah. Sorry, I should have explained. So one is the top view, one is the bottom view, or one is the top view side. They're just two different views of the same process, essentially. Sorry, this one? That one? I don't know what it is. Sorry, but yeah, yeah. Okay, so for instance, these morphogen gradients give a precision of a single cell, right? With a single cell precision over here, the fly can say, hey, this is going to become the head and so on, right? But then there's something about that gradient and that point which drives the flow, right? And so what drives those flows are the fact that there is consumption of energy at these small scales, all right? And I'll come to that in a little bit. Okay. All right, so how I want to think about this going forward is, instead of thinking about structure, function, behavior, reproduction, and so on, that we normally define these biological things by, I want to think of, physics things, and then see how far we get with that. This is the simple-minded picture that I originally told you about. All right, so let's start with physics then, right, about matter, and then slowly build on from here. All right. So we all know that the interaction energies between the constituents of a gas or of matter give rise to the phases that we see, or the macroscopic behavior that we see. So for instance, if the interaction energies are lower, much lower than the thermal scale, you get a gas. If they're comparable to the thermal scale, you get a liquid and then a solid, right? We know a lot about these things, and there's something in between these which operate. There's some stuff. Let me just call it stuff for now, which operates in these energy scales in the KBT regime, where the interaction energies are comparable to the thermal scale, right? And this is the stuff we're all made of. Things like lipids, proteins, macromolecules, DNA, all so-called soft matter, right, is what falls in this regime, right? And like I said, it's characterized by interaction energies which are comparable to thermal energies, which means that most of the processes that we run happen at room temperature. The assembly happens at room temperature, and so on, right? And still we are not somehow rigid like a crystal. So the soft matter is what I'm going to tell you about, right? And in particular, I want to draw your attention to a class of materials known as liquid crystals, right? They're aptly named because they sit literally between liquids and crystals. So these are comprised of rod-like molecules. So you can literally think of this as a liquid in here, a liquid which is comprised of rods, instead of balls like this that we're used to thinking about gases and so on, right? So what this rod-like nature of this individual element allows is that it can exist in increasing degrees of order. For instance, all the rods can align their directions without any particular ordering of the positions of them, okay? This is a so-called nematic state of this liquid crystal, okay? You can increase this order further by having all of them point in the same direction and have stacks of them, but within the stacks, there's no crystalline order. Within the stacks, they're like that, right? So this is a material which is solid in one dimension and liquid in two dimensions, right? So this is definitely more ordered than this. And eventually, if you keep going down this way, you'll hit a crystal. There are other exotic phases. And so you can see that the interaction energies are essentially increasing in this direction, okay? So why I wanted to bring up these liquid crystals is because of this, right? The moment you see a school of fish, it reminds us of this nematic liquid crystalline phase. They're really just rods which are sitting in this blob, all right? But there's a difference between these two in the sense that if I want this to do anything interesting, I need to drive it. I need to provide it an electric field or a shear or something like that, right? For it to flow or for it to function in my watch. And so this is what we call as passive matter, where we have to drive it at the boundaries. Whereas this one over here, each individual element is capable of consuming and dissipating energy, right? So this is the distinction that we make between matter and active matter. Active matter is something that's comprised of units that are consuming and dissipating energy, right? And just this, what may seem like a subtle difference, gives rise to a lot of interesting physics on the one hand. And if you take the physics of these guys and tweak it a little bit, you get surprising similarity to what these things do, okay? And so this is not restricted to fish schools alone, right? So as you might have already imagined, this is very similar to fish or to birds. So for instance, a bird may not really look like a rod, but then, yeah.
There's life and so on, which is very complicated. Then there's the gas, solid, which seems simpler. So we don't have a very good understanding how matter, life comes. So then from the kind of matter, we can even go further down, right? Like quantum mechanics. From quantum mechanics, we don't have a very good understanding of how these macro states come. Is that correct? What is that?
Right. So I would think that at every level you have these emergent properties which preclude you from just getting a description from bottom up, right? And in this case, all I want to say is the similarity in these structures and what we can and how we can make use of that knowledge in trying to understand these things. I'm in no way trying to go towards building a description from bottom up of the fish. Right? All I want to do is say that this fish is something like this rod with a slightly different property in that it is consuming energy and so it can self-propel. Okay, so let's work. Yeah.
The second question is just curious. So you said most of life exists in the liquid crystal. Like for example, we don't have a gaseous life form. We don't have a liquid life form.
Right.
Why does life exist?
Again, I would think that you need structure of some sort, right? So you need to maintain structure. You just don't want us to I don't want to be occupying this entire room. I want to be here, right? But at the same time, I want to be able to do this, right? There's some delicate balance of maintaining structure and maintaining flexibility, if you will, right? And so that may be one reason why things may operate in this regime. But yes. So as you go down, you have different that can capture this ordering information, right? Different. Well, I don't know what you mean.
It's a measure of disorder.
Yeah. So this is what captures something like that, right? Okay. Yeah.
So I think in the 70s, a guy called Rennie Tom, he was something like catastrophe theory. He was a physics scientist. And there's a book he wrote about explaining some of morphogenesis using catastrophe theory. I don't know, Ravi, I don't know if you've heard of that. Heard of it? So there is a little bit of this that he tried to explain, but I don't remember the details of the book, but there's some interesting nuggets in there which have been discredited. I mean, some have not been discredited. So I don't know if you.
I don't know about this, okay, but so I'll be happy to.
Okay.
Yeah, we can. Yeah, I don't know.
We have some of.
I see. Okay. So that's good. Okay. So here I want to point out to you that even at this very small scale, bacteria. Sorry. Okay, so I'll touch upon this very briefly. It turns out that every bird doesn't need to know about every other bird, right? It has even been shown that each bird tracks something like 7 neighboring birds, and then even that's just sufficient for them to be cohesive and get this large-scale flock. Okay, yeah, so I just want to point out to you that these phenomena happen at various scales, right? This is bacteria, each of which is a micron long, and they also show similar collective patterns. And this is a movie, the fluorescent blob here is comprised of actin filaments. These are nanoscopic elements which sit in our cells, and which is what allows us to do this, flex our muscles, for instance, right? So what these are little filaments along which motors walk, okay? And it's this, again, the consumption of energy, right? So you can think of this as an element which consumes energy and moves. Right? This one here?
No, the one on the lower right.
Ah.
What is the log? Is that the walker's concept?
Yeah. Yeah, so I should do this more carefully. So there's a substrate, a glass, onto which motors have been attached, and then there's this actin filament. Okay.
In this case, walkers are stuck to the glass.
Exactly.
And walking, they're sort of moving.
They're just doing that, and this guy is just moving along, right? And if I look from the top, I have a filament that's just doing that. Many of them are doing that, right? And much like the fish or the birds, they're forming this group.
And here they're coupled because they're bumping.
They're coupled because they're bumping into each other. Yeah. Okay. So that's. Thank you very much for that point, because that's also what we want to do here, where, of course, these fish and birds have brains and senses, and they're processing a lot of information. And the claim here is that we can, at first approximation, ignore all of that, and then still say something meaningful across these scales. All right. Okay. So now I just wanted to quickly go over a very simple model that can give this. This was developed originally by a computer scientist, Craig Reynolds, the so-called Boyd's model, where there are individuals who are moving, and he had 3 very simple rules. One is alignment, cohesion, and some repulsion, so that they all don't sit on one big spot, right? So essentially what this is saying is that if I'm somewhere out here, I will try to move towards the group, that's cohesion. I maintain a certain distance from my neighbors and I align, right? And this already is, these 3 simple rules are already sufficient to give something that looks like a flock, okay? Yeah, so they just move towards the center of mass, exactly. So this was somewhat, I hope I get this spelling right, in 1993, I think. It's essentially the same thing, where he writes, so now this is defined over some neighborhood of each individual bird. It's not a mean field, it's not, every bird is not seeing every other bird, but there's some radius in which the birds are copying each other. So this is really like the Ising model, except the spins are not fixed, they're moving. All right? And there's some noise with which these guys copy each other. All right? And so this model has been studied extensively now in the context of these flocking behaviors. And it shows a transition from, so if I have an order, which is basically just an average of these guys, there's a transition from disorder to order, which depends on this noise or the density. Right? So this is also 1 over density. Okay? So now the attempt is to think of continuum versions of this and think of these things as fluids. Okay? Sorry, so that's density. So it's just the number of birds that there. So if I increase the number of birds per unit area, so yeah, unit volume, you get that as well. Okay. So in thinking about continua, I just wanted to quickly introduce you to the Navier-Stokes equation. Probably all of you, or many of you have seen this, but I just want to point out to you the features that are interesting over here and go further, right? So this is a description. This is an equation that's describing the flow when you apply a shear to a material or a fluid. So you develop a flow field in this material, so the nice thing about writing this equation like this is that you separate out what constitutes an inertia. So if you see, this is really rho is the density, right? So this is momentum, and that's a momentum flux that's being carried through the flow, and that's the stress. So that's the pressure gradient that you apply. That's the viscosity, and so you have a viscous dissipation here, right? So all this is really Newton's laws of motion, F equals ma, right? And this goes together with a condition normally that's written for incompressibility. You say that the divergence of this flow field is 0. Okay. And now I want to non-dimensionalize this and use this non-dimensional form to get some intuition into the small scales or the bacterial scales that I'm going to talk to you about. Okay? So when we non-dimensionalize this, we get out a non-dimensional number, a so-called Reynolds number. Okay? It's the ratio of density. U is the characteristic speed of the flow. L is the length scale, and eta is the viscosity of this fluid. All right, so where does this Reynolds number come from? Unsurprisingly, it's named after this person, Osbourne Reynolds, and he performed an experiment where he looked at the flow through pipes, right? So he had a pipe, and he had a little vial of dye, which he passed also through this pipe, right? And by visualizing this dye, he was able to say something quantitative about the flow, nature of the flow. All right? And what he did was to vary the size of this pipe, the pressure head, which basically changes the flow speed or the flow velocity through this pipe, and so on. Right? And this is what he found. He found these two regimes. One he called laminar flow, where everything is linear, so this little string of dye that's passing through the fluid goes through unperturbed. And then there's this very familiar turbulent regime. All of us that have been in a plane know this, right? Where this little stream of dye breaks up into eddies and vortices, right? And it gives rise to a large-scale mixing of the flow. And these regimes are related to this Reynolds number. This Reynolds number place these regimes somewhat depending on its how high it is. So if the Reynolds number is sufficiently small, you get these linear flows. And if it's sufficiently large, you get these turbulent flows. All right? So going back over here, if the Reynolds number is sufficiently small, it means that inertia is weak compared to viscosity, right? So this is a regime in which the viscous forces dominate the proceedings, right? And in such a situation, you can essentially get rid of all of this, and then you get a flow field that's just dependent on the gradient of the pressure that you apply, right? What you will immediately notice from here, this is the so-called Stokes equation for the flow. What you will immediately notice over here is that it's no longer time dependent, right? This is saying that if I have a flow in this direction, it might as well be backwards in time, right? We lose time dependence. And this is a beautiful demonstration of this reversibility, right? So let me just pause this video and tell you what's going on. This is a famous video made by G.I. Taylor. So there are two cylinders over here. One cylinder inside another, right? And the gap between them is filled by a viscous fluid. And this has a lever which can turn, which causes a flow in here. What he will do at some point is, with a little syringe, put a drop of dye, and then create a flow. Okay? That's that drop of dye. Now he just turns it back. Yeah, now he'll start to turn it back. Quite remarkably, it all comes back together, right? So it's really time reversible, right? And so looking from the top, so he'll do this from the top. Now you can see why this is called a laminar flow, right? So he'll make a little square over there and then repeat the process. And then if you watch every point along this thing, you will see that each point is just moving in a line. Right? So literally you're getting little sheets of flow, little laminae of flow that just go together, right? I can watch this like forever and still be amazed that it's so beautiful. All right. Okay, so what I just told you is that in this Stokes flow regime, it's a viscosity that dominates the proceedings. You can think of yourself being immersed in toothpaste, and that's how it is, right? These flows are linear and laminar, and they're time reversible, as we just saw. Okay, so now I want to go into what this means for life at the very small scales, for bacteria, and what this does. First, I want to show you a video of this thing called a scallop and how it swims. And it's swimming away, right? And what this once it stops doing that, it just stops moving, basically. So what this guy is doing is that it's literally, two arms with a hinge. And then he's doing that. And that's propelling him, all right? So what this flapping motion does, we just learned that if this was happening at the small scale, everything is time reversible. So if I do that, I move forward. And if I do that, I just move that much back. So it'll never work at the small scales, right? But it's working here because this flapping is causing turbulent eddies at the back, right? And it's actually an inertial propulsion mechanism, okay? So this would never work in the small scale, and so this is the so-called Scallop Theorem, where it says if you just have 1 degree of freedom, 1 hinge, that's not sufficient. You need either a time reversible, or let's say if these were flexible, and then you get a time irreversible motion, then you can move, right? Or you need 2 hinges. Okay. So now I just want to very quickly point out to you that this Reynolds number is about 10 to the 8 for a whale, 10 to the 4 for us, and for a bacterium, it's 10 to the minus 5. Right? So we all see the same water very, very differently. Right? For us, we are able to make use of flailing about and moving, whereas a bacterium cannot, because it has to fight these irreversible flows or reversible flows. And so it comes up with very interesting mechanisms to swim. In this case, in the case that I'll just tell you about, it has a flagellum, which is like a corkscrew, and then it does a little corkscrew motion, and so that motion is time irreversible, and that helps the bacteria move. So these two are the same, because it's the same water, the density. So a characteristic speed for a whale would be 10 meters per second, something like that, and that is 10 meters, right? And so that number for water is about 10 to the 6 or so, and you get that, right? Whereas for a bacterium, it swims about a micron per second, and it's about a micron big, and so that's the number, right? So it's really a matter of scale or size. Okay, so that's how this E. coli swims. This is a fluorescent image of the body and flagella that are glowing bright, and so you can see that it's doing this corkscrew motion, and it's moving along. But then you'll also see something remarkable that happens every now and then, that what appears like one flagellum breaks off into many. So it's really a bundle of flagella, right? So they all come together, and then they do the corkscrew motion, and the bacterium moves. But every now and then, it just lets go. And what that allows the bacteria to do is, it allows it to reorient, right? So what the bacteria does is a so-called run and tumble motion, right? So it runs when the flagella are all bundled together and it's swimming straight, and then it lets go. It does a tumble, which basically randomizes its direction, and then it goes off in that direction, right? So a bacteria really does a random walk, okay? But how does it ever get anywhere when it wants to? Is because it can chemotact, or it can bias these runs and tumbles, right? So if it, for example, has some food up there and it wants to climb up that way, every time it's moving up the gradient, it keeps going for longer, right? But then if it's going away from that, it tumbles more frequently, and then it reorients its time, and so eventually it moves up the gradient, right? So it biases its random walk and then moves up, right? So this is a very clever trick that the bacteria uses, and it turns out that it's actually computing this gradient, and doing something quite sophisticated with the molecular machinery below. Sorry?
Is the tumble essentially random orientation, or is there some?
So in this case, in both cases, it is a random orientation, right? Because the rotational diffusion takes over, and then it's just pointing in some direction, and then it goes off. The chance of that is, of course it can, but it's a very small chance, right? It's a random walk. Yeah, more often than not, it'll, yeah. Okay, now that I've set up all these things, I want to show you 3 surprising aspects of this activity, right? Because I want to get to you, convey to you the essence of how this activity at the level of the individual makes things different. That's so the first experiment that I want to tell you about is bacteria in a box. Okay, I just told you that each individual bacterium does a random walk. All right, so now if I take a bunch of bacteria and put it in a box, they're just essentially gas molecules in a box. Okay, so now this box is separated into 2 halves by a set of walls with gaps inside them. Right, so there's a membrane which separates these 2 things. Right, so you would expect that really nothing interesting happens. So if I put all the bacteria this side, eventually I will get a uniform or homogeneous distribution of this bacteria in this entire chamber. Right? So if I look at the ratio of the number of bacteria this side to that side, just stays at 1 if I start off with a homogeneous distribution. All right? So now, the same experiment with a small difference. The walls that separate the 2 halves are no longer symmetric, but they're asymmetric. All right? And if you start with the homogeneous distribution of bacteria, the bacteria find themselves in one side of this chamber suddenly. Okay, so and if you look at the ratio, again, this goes up. Okay? So I just told you that all of the flows, the swimming, everything is happening in this linear regime. Everything is time reversible. But then there's something about this asymmetry and the fact that these guys are propelled, which breaks this time reversibility, right? Okay, so at first glance, this appears like the classic Maxwell's demon. The demon, the devil, of course, lies in the details. Yeah. Yeah. I want to say yes. I don't, because I don't know. I think it's, yeah. I don't see any reason for. Yeah. Right. So the devil, of course, lies in the details, and I'll come to that later. And of course, you can now use this to do some useful work, right? So if you take little gears and put it in a bath of bacteria, you can get rectified motion. So you can really drive a little pump with this bacterial fluid. Of course, you can't do this with a gas, right? So Feynman proposed this ratchet mechanism, which of course, as you all know, doesn't quite work, right? And so there's something interesting about this active fluid that allows one to get this out. All right. Second surprise is that if you take a little sheet of this bacteria, so now here you're watching a movie of bacteria which are confined between 2 plates, and you already see whirls and vortices in the motion of this fluid. And if you carefully analyze the vorticity in this flow field and get out the energy spectrum as a function of the wave number, you'll really get turbulence. All right? So these exponents characterize turbulence as we know it in clouds and in the ocean and so on. So you're getting turbulence of a very, very similar sort at these small scales where I just told you flows should be laminar. That's the second surprise. Sorry? Okay. So these exponents So if you didn't have, first of all, you would not expect any sort of inhomogeneity in the flow, right? So there's no reason to expect that you will have any patterns in the flow at all if you didn't have this mixing. This is the third surprise. So if you take this fluid, so let's say I take bacteria and suspend it in water, right? So water has a certain viscosity that you can measure. Right? So now I'm adding some particles into it. At first glance, you would think I'm adding something to it, so it must become somehow thicker, right? But then there are also counterexamples in our everyday life, like ketchup, where it is thick, but then when you apply some shear, it actually thins and flows out, right? So what I'm showing you here is a measurement of the viscosity of a suspension of bacteria as a ratio to the viscosity of water, right? As a function of the amount or the fraction of bacteria that we put into the suspension. So when there's no bacteria, of course, it's the viscosity of water. And as we increase the amount of bacteria, the viscosity decreases. Okay? That itself is not a very great surprise, like I just told you, ketchup thins. But the real surprise is this. You're getting a superfluid. The viscosity of the suspension actually goes to 0.
On purpose by spinning their flagella that way and preserving their orientation. So you're saying when you do the numerical measurement of the viscosity based on the motion, you see this momentum dominated.
Exactly. Exactly. Okay. Exactly. So yeah, I couldn't have explained it better than what Chris just said, that this is really a momentum-dominated regime, as we know it. And you're getting, again, this at these linear regimes that I just talked to you about. Right? So that's the third surprise. And so what I'll also very quickly do, maybe not. So I'm actually going to end my talk here, even though I have 5 minutes. We'll take questions, and I'll resolve these puzzles when I come back and point out to you the role of activity, and then go on. But, yeah.
So this bacteria is doing the tumble run thing that all these bacteria are doing the tumble run.
Every bacterium left by itself is doing this run and tumble. Yes.
And the tumble is reversible. The run is reversible. The tumble is also reversible.
So the tumble essentially randomizes, right?
Correct.
So
but if you, it doesn't create any turbulence. It doesn't do
no. So it is like, so it's an overdamped motion, right? Yes. So you want to think of each of these events as where you're picking the orientation from a distribution. Yes. So this one is, but not this, right? Because there's some biasing that goes on over here. Oh, I'm sorry. I'm really sorry for being so shoddy. Yeah, also these, and the noise too, right? So there's some direction. Sorry. There's no food. So none of this is, okay. Of course, food is necessary to keep the bacteria going. Otherwise, they'll die and nothing interesting happens. But other than that, food is not playing any other role in.
So the question is that the bacteria will move only for this.
Why do bacteria move around? In search of things, I guess, yeah. There's food.
Yeah, the point is that there might not be a gradient.
Sure. If it is a well-mixed, yeah, of course, if it is a well-mixed thing, in the lab, they're treated very, very nicely, you know, they're given abundant food and they don't need to do this, but not.
In that case, they should only tumble. They should do that.
They probably don't even bother to swim in the lab. They just probably form biofilms where they're essentially forming little cities and then just sitting in. Yes, sorry. In the case where it's superfluid, the bacteria are swimming, right? And so they're, okay. So the point you're trying to make is that there is... Yeah. Okay. So you can always get bacteria to live, I mean, not die and swim, you know, and these experiments are done in these conditions, where, you know, you can keep them going for a while, but then they're not so happy that they're just doing nothing.
I'm asking because there are like genetically modified tumblers and swimmers, right? The ones who lost their ability to do one of those things. Did they use one of those things to only swim?
So these guys swim normally. They have a flagellum, and so what I described to you, the E. coli movie that you saw, that's what's happening here as well. Yeah. You had a question? No. Sorry? Here? Oh, you mean how long these flagella are? Yeah, I guess the speeds and so on will depend on that, but not, you know, the dynamics will not change too much.
So just to make sure that I got the paradoxes of the thing. So they have this irreversible corkscrew motion.
Right.
The paradox is they all end up on the right side.
Right. Right.
But you sort of set up this kind of lobster trap thing where
This.
you go from this side, tunneling.
So if this was gas, still, even though, so I cannot create asymmetric barriers in this room and create an air condition or a cooler, right? I cannot do that. So there's something else going on over here, right? That's, yeah.
Second paradox is that they're generating turbulence even though they don't
So what I just, yeah, exactly. So you're getting something which is inertia, a regime which you normally see where it is inertia dominated. That's happening in this regime where everything is viscous dominated, right? I will point out to you that, you know, there's maybe, so there's something about, so you want to think also of this thing that I told you, which is also relevant here, where things are aligning with each other, right? So things are aligning, and it's a fluid which is comprised of things which are aligning, right? And so it's an instability in that fluid, right?
last paradox was
Well, the fact that, you know, there's a change in viscosity and yeah, and the superfluid nature is like a really big surprise. But even the fact that, you know, you can get reduced viscosity or increased viscosity is already quite interesting.
what Chris said didn't quite explain that paradox.
No, he just described the nature of the state.
So if you measure the viscosity by putting a spoon in the water yourself and swirling it around, in that sense,
Right.
that motion would not necessarily exist.
Right, exactly.
But if you watch the bacteria and see where they go,
Exactly.
They're floating around as if there was no viscosity at all.
Exactly.
But this is sort of because
Well,
of their software, as it were. I mean, if they had evolved to, well, not just, I know it's also mechanical forces, but if they had evolved to care more about what direction their neighbors were going in, and then they would see if they were getting close to their neighbors, then they would look like a viscous flow.
Right. Right. So presumably there's something evolutionary that drives some of these behaviors, but there's a large mechanical component that gives rise to them as well. Sorry, you had a question.
The viscosity are going negative, the rate.
Yeah, it's actually going negative. It means that you have a pump sitting in the fluid.
So they exhibit like the rate that liquid emissions climb up the water.
Right. So that exactly, that shouldn't be too surprising at first glance, right? That, I mean, if I have a jar of ants, they can climb out, right? All right. So I think I'm going to stop there, and then for the next half, I'll start with addressing some of these things and tell a little bit about evolution and so on.